Good to see you all. I'm Wook Hur from Yonsei University College of Medicine. Today, we're going to talk about how we develop the specific peptide variant and the efficacy of such peptide variant. We applied a series of patents, one in Korea this year as regards TAT peptide variant, and another in the States as regards TAT peptide in 2019, the latter of which was registered. Here's the key concept. In the disease's standpoint, obesity is as serious as causing accumulation of additional lipid in the body and causes high blood pressure, cardiovascular diseases, and metabolic disorders such as hepatic cirrhosis and diabetes. One another reason we're interested in this study is the idea the slim disease, oftentimes found in African populations, is caused by viral infection, like HIV infection. So we've built up a hypothesis that TAT, as the most important transcription factor that impacts copy and transcription of HIV, decreases adipose tissues. And under that hypothesis, discovered that recombinant TAT protein effectively inhibits obesity when administered by obese animal models. We went on to also discover that by mapping the specific part in TAT polypeptide that has anti-obesity activity, such anti-obesity activity is originated from TAT38 peptide 20 to 57. Despite such an advanced TAT marker, we thought that the physical properties of TAT38 still need improved, which is why we went on to developing a variant of TAT38 and referred that variant as XFAT. With a lot of drug in the anti-obesity drug market accompanying a variety of side effects, a highly potent drug with less side effects are in need around the world. Here's how TAT is made up in terms of amino acid sequence. In most studies that have been conducted so far, TAT is comprised of a total of 101 amino acids, started off with the protein-rich domain, and followed by cysteine-rich domain, core, arginine-rich basic domain, glutamine-rich domain, together generating the anti-obesity effect in recombinant TAT protein. When using recombinant TAT protein, weight loss occurs highly adipose specifically. When TAT is administered by obese mouse model, a huge decrease in lipid was observed, as evidenced by the reduced size of adipose tissues that we're going to take a look at later. Reduction in size of adipose tissues are observed both microscopically and by way of scanning electron microscope, and is specifically induced by triglycerides that overexpresses TAT. According to DEXA scanning, we see a significant decrease in fat mass while lean body mass is the same. When it comes to obese mouse models, they have very fatty livers and, from time to time, liposteatosis, as you can see in mouse model 4 in the bottom right. And you can also see liver gets cherry red and becomes healthy by the effect of TAT. Same thing for TAT38 we have mapped. As you can see in the figure C, weight loss is very evident. And as you can see in the figure F, adipose tissue size has decreased very severely, while inducing rapid absorption of glucose and enhancing insulin sensitivity, and decreasing blood sugar level, of course. Next, when we tested TAT38's inhibitory effects against adipocyte differentiation and adipogenesis, as you can see in Control A, differentiation began on day 2, followed by generation of adipocytes on about day 4. On about day 8, adipocytes are fully differentiated into adipogenesis. However, TAT-treated part adipocytes were still not fully differentiated on day 6, ended up synthesizing very small amount of lipid on day 8. Take a look at those day 6 figures to see TAT treatment effectively inhibited adipocyte differentiation and adipogenesis. All in all, we now know that, at least in culture media, TAT38 effectively inhibits adipocyte differentiation and adipogenesis. In numbers, the TG content on day 7 implies that about 70% of lipid was inhibited from being synthesized, which of course is very potent, despite low solubility in water and poor synthesis. So we went on to develop a variant to improve the solubility in water and synthesis. It is to note here that the fact that TAT as a transcription factor comprises cyclin T1 CDK9 complex was published on Nature. Taking a close look at the backbone of TAT on the figure A, the very thin lines and red and purple strands are basic domain, cysteine-rich zinc, and finger-like domain, respectively, each of which interacting with cyclin T1 and CDK9, respectively. These two domains are linked by linear peptides. If we take a closer look from behind, it gets clearer. So, in terms of distance, if the distance is similar to the distance of alpha-aliphatic acid or the octanoic acid, then it counts, as we thought. So, in our design, we've discovered a total of 11 peptides and substituted them with octanoic acids. Out of the thought that the effective anti-obesity effect can be resembled based on the structural similarity, and that was very true. So because a total number of peptides is 228, leading to higher yield, easier synthesis, and improved solubility, we went on to treat adipocytes with TAT and perform staining to see how adipogenesis is affected, and we found out that it virtually was shut down at about 10 micrometers, while also effective in 1 micrometer, which is why we thought that it is effective at around 3 micrometers on average. As you can see in this figure that reproduces the results, the effect was observed at both 1 micrometer and 10 micrometers, although depending on microscopic field, and the effect was superior in 10 micrometers. This is just another way to view the effect is very superior in 10 micrometers. Next is adipogenesis assay, result where XFAT was also superior to TAT38 in our assay conditions. When oleic acid was added in assay, inhibitory effect against synthesis of fatty acid in hepatic podocytes was potent starting 3 micrometers and up to 10 micrometers, evidencing synthesis of fatty acid can effectively be controlled in adipocytes. This figure represents the effect of XFAT in disease model mice called DIO mice, meaning diet-induced obesity mice, where obesity is induced by high-fat diet, where such high-fat diet was changed to normal chow diet when tests began. To see more accurately and clearly the effect of XFAT, according to our test where intraperitoneal injection of XFAT took place, after a month-long treatment with reagent, weight loss was observed by approximately 10%. As for food intake, the XFAT group mice took a bit less food than the control group mice, with statistical insignificance. The black dots represent results from the XFAT group mice. One other thing to note here is, although not statistically significant, food intake was also controlled in the XFAT group mice. While we did not take it seriously as it was not statistically significant, I think it's worth mentioning. Next are GTT and ITT. Although the GTT results are slightly different from the ITT results, as blood glucose level is generally low in the GTT results that represent faster glucose absorption, while insulin sensitivities between two groups were not quite distinguishable. Anatomically saying, decrease in adipogenesis was very evident in the XFAT group mice treated with TAT. It is to say that 10% change in TAT actually means a lot. With smaller, yellowy inguinal white adipose tissues, where we can reasonably assume that decrease in adipogenesis takes place quite naturally when treated with TAT. Smaller bat tissues are also notable. Anatomically saying, decrease in the size of epididymal white adipose tissues was also observed, with even more evidence decrease in lipids observed in bat and liver. Mechanistically saying, expressions of those genes related to adipocyte differentiations and synthesis of lipids, such as SREBP, PPAR-gamma, and CEBP-beta were low. All other results except for PCSK9 were quite satisfactory. In other words, however, further studies to improve this PCSK9 results need taken place. That is all. Thank you.